INTA in Argentina is the National Institute for Agricultural Technology. Alfalfa, also called lucerne, is a perennial important forage crop in many countries around the world. Its primary benefit is the combination of high forage yield per hectare and high nutritional quality. The National Alfalfa Breeding Program is coordinated from the experimental station of INTA Manfredi, located near to Manfredi in Córdoba province, 75 kilometers southeast to the capital of Córdoba. In terms of development of commercial cultivars, since 1987, INTA established a policy that we can have joint ventures with private companies. Since its creation in 1970, INTA has registered a total of 18 varieties of alfalfa from fall dormancy 5 to 10 at the National Register of Cultivars of the National Institute of Seeds. The program developed around 40 experimental populations each year, called CIMAS Sport Synthetic Manfredi. Argentina, with more than 2.5 million hectares, is the second largest alfalfa producer in the world. Approximately 80% of the total alfalfa area is cultivated under rain-fed conditions for dairy, beef and hay production in the Pampas region, while the remaining 20% is devoted to hay and seed production under irrigation in the northwestern, western and Patagonia regions. 60% of the total area are planted as pure stands primarily used for dairy and hay production, and 40% in mixtures with temperate grasses, mostly utilized for beef production. Hay production for both domestic and international markets is becoming increasingly important. Average forage yields are about 15 metric tons of dry matter per hectare per year, and vary greatly according to soil and climatic conditions, management practices and moisture availability. In rain-fed conditions, productions can go from 11 to 27 metric tons of dry matter per hectare per year. Argentina has now in the market about 100 or 120 cultivars, which is too many. In order to help the farmers to select or to choose cultivars, INTA is conducting since 1990 the National Alfalfa Cultivar Evaluation Network under cutting conditions in many locations, about 16 across the country. The main idea of this trials is to group the cultivars in intermediate fall dormancy and without fall dormancy or non-dormant. The trials are conducted for 4 years. During the season, data is collected, forage yield, production in dry matter at 10% blooming and persistence, and analyzed. All the data collected of the varieties that the companies want to evaluate on the network are summarized and is made available annually at the INTA's webpage in Avances en Alfalfa publication. In Argentina, alfalfa is mainly used for animal feed associated with beef and milk production. Since 2002-2003, with this period of very high prices of grain, most of the good soils in the Pampas region in Argentina are devoted to grain production, so the forages in general and alfalfa in particular were moved to soils which are not so good all the time. Direct grazing is still very popular for animal production. However, the use of forage reserves such as hay is increasing, accompanying the changes in the systems, increasingly confined or intensive and with a greater contribution of supplementation. We have two different big environments. One is with good soils in which our dairy production is located and they are using alfalfa, so the breeding goals there are high forage yield, persistence and high quality. For those marginal environments with not so good soils, people are asking for tolerance to abiotic stresses, particularly drought and salt. The objective to release commercial alfalfa cultivars at INTA's breeding program is carried out with the financial assistance of the company Palo Verde through a joint venture since 2009. We are in charge at INTA to do the selection process and the evaluation process, and we registered the cultivars under the property of INTA. And our partner Palo Verde is responsible of the seed increase, the commercialization of this varieties, and also pay to INTA for our operations costs and a royalty of this varieties that they sell. In order to develop varieties adapted across the country with very high yield potential and high forage quality due to multiple pest resistance, we are concentrated our effort in fall dormancies 5 to 10. Among the main pests that affect alfalfa we can mention The root curculio complex, the alfalfa caterpillar, the thrips and the aphids such as the spotted aphid, the blue aphid, the green aphid and the cowpea or black aphid. Among the main root and crown diseases we can mention Phytophthora rot root, anthracnose, fusarium wilt, root and crown rot complex and corky root. The main leaf diseases are common leaf spot, yellow leaf blotch and rust. With Palo Verde, we were released 3 cultivars, Pulmari with fall dormancy 6 and Traffle and Lime with fall dormancy 9 and 2 more are just finished the registration process. These are the characterization, fall dormancy, Phytophthora, anthracnose, fusarium wilt, and the aphids blue, pea and black. Pea aphid is in USA and green aphid is in Australia. When we talk about high resistant, it means at least 51% of resistance. We have 2 new materials registered in 2020. This is Amaya with fall dormancy 10, selected to have a high multileaf expression, and Cumin with fall dormancy 9, which is a salt-tolerant variety. At the table, you can see the characterization in terms of fall dormancy and resistance to diseases in aphids. Amaya have more than 3 leaflets per leaf. We were able to increase the multi-leaf expression from 6% to almost 80% in the 4th cycle of selection. This gives us a significant increase of the leaf and stem ratio compared to the base population, and also the crude protein content was also significant and high after 4 cycles of selection. This variety is intended primarily for high-quality hay production. Cumin, the salty-tolerant variety, were selected in these salty places of Isla Verde in Santiago del Estero and some populations called SISA for synthetic Santiago were evaluated in the salinity lab at Riverside, California, and were performing a lot of testing. And those are the data. And you'll see that Cumin were performing very well in 2 types of salinity conditions. One is sulfates and the other one is chloride. Everything presented so far is based on conventional or traditional breeding, where plant breeders use the natural genetic variability of alfalfa for the development of new varieties. On average, the period from the initial selection of plants and the registration of a new variety takes from 8 to 10 years. Our general scheme that how we developed a new variety is like that. We used 3 different methods. One is the phenotypic recurrent selections at greenhouse or field selection. For field selections, this is the type of plants we are usually looking for with wither crows, high number of stems, healthy plants and other good agronomic characteristics. Under field selection, we select with absence of corky root disease and the complex of rot and crown diseases, which is a complex or combination of different pathogens. We don't have any resistance to these 2 diseases or any protocol. Also, we discard plants with leaf diseases. This other is a viral disease, a complex of at least 7 virus. This is a particular disease in Argentina and is very common all over the country. You are able to see the symptoms after 2 or 3 years. This viral disease that causes stunting, evident decrease in the height of the plants, yellowing of the ribs and severe deformation of leaves, including wrinkling, epinasty and thickening of the ribs in the underside of the leaflets, originating papillose formations and enations of different sizes. The selected plants are going to the pollination cages. We put some honey bees, and after pollinations and seed formation we collect the pre-basic seeds or synthetic one. The other 2 methods are strain crosses combining different cultivars, and we also use selection protocols to increase the level of resistance to aphids and diseases. For the disease-resistant selection, we use the protocols defined for the North American Alfalfa Improvement Conference. You can look at the NAAIC website in which are the protocols or standard tests. In this particular case, this is the protocol for Phytophthora root rot. We plant the materials we are going to select in lines and intercalate checks or controls, resistant and susceptible controls. We use 3 reps and the controls to check if the inoculum is more or less even spread on the field. This is a disease that need a very high level of moisture, so we just irrigate many times in order to promote infection and the development of the disease. And after 6 months or a year, we take out the plants, wash them and read the symptoms. It's a very laborious and tedious work. We use a scale to read the symptoms from 1 to 5. 1 and 2 are resistant plants and 5 are susceptible plants. The selected plants are going to the pollination cages. In the case of anthracnose, we use pots to sow the experimental populations we are looking for resistance. We have one row which is the resistant control and one row which is the susceptible control. We cultivated the fungus and do the dilution of spores in a known concentration to spray on the seedlings. After the spray, we put the pots 2 days under moisture conditions to develop the infestation, and after 15 days we read the symptoms. The resistant plants are selected and transplanted to the pollination cages, and this is what we call one cycle of selection. For aphids is similar. We raised the aphids on alfalfa stems under control conditions. We do the same thing here in pots like I mentioned in anthracnose, the resistant and susceptible control, etc. After 15 days with the right number of aphids feeding on the plants, we eliminate them and read the symptoms and transplant the resistant plants to the pollination cages. So those plants are coming from field selection, or the selection from Phytophthora, anthracnose or aphids. We tried to have between 60 and 70 plants to conform a pollination cage to prevent inbreeding depression, which is very important in alfalfa. After pollination, we collect the seed and we do some seed increase of this experimental populations in other zones more appropriated for seed increase in terms of dry climate and irrigation, and obtained the synthetics populations 2 or 3 to use these seeds for evaluation process at different Argentinian locations. We evaluate under cutting conditions which are the best materials in terms of yield, production, persistence and other variables. This best synthetic populations are presented to our partner to decide which and start the registration process for a new cultivar. Also, we have other breeding goals in terms of abiotic stresses tolerance, drought, waterlogging or salinity. B. Bloat tolerance, near-infrared spectrophotometry, and C. Biotech, diseases resistance and herbicides tolerance. Also editing. In terms of biotech developments, we work collaborating with the CONICET INTA in Castellar, Buenos Aires. For drought tolerance conditions, we start a selection program with 142 half-sibs families coming from Italy. We evaluated them under drought conditions at INTA Santiago del Estero, and we were able to select 200 plants with good performance and combining them in a pollination cage to have seed for drought mechanisms evaluation. Also, at INTA Rafaela we are performing a selection process looking for water logging tolerance under control conditions. In terms of bloat tolerant, because grazing still important in Argentina and bloat is a big problem, we are looking for lower initial dry matter disappearance using NIRS technology. After 2 cycles of selections, we are now performing an evaluation of the population, testing the bloat performance in sheep under grazing conditions. For seeds provision, we recommend seed companies that offer the guarantee of the genetic and physical quality of the seeds.